Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore coenzyme in respiration. At the end of this lesson, you will be able to describe the role of coenzyme A in the transfer of acetyl groups, as well as describe the role of NAD and FAD in transferring hydrogen to carriers in the inner mitochondrial membrane. Let's get started. So a coenzyme. So what is a coenzyme? Coenzyme is a small organic non-protein molecule that assists enzyme by transferring a particular or a specific chemical group between reaction. So it acts as a cofactor for enzyme, but they do not form a permanent bond with the enzyme structure. So the coenzyme involved in aerobic respiration includes coenzyme A, nicotine amide adenine dinucleotide, NAD. So this is the full name of NAD. But in our syllabus, we are allowed to use an abbreviation NAD instead of a full name nicotine amide adenine dinucleotide. Then how about FAD? So FAD is known as flavins adenine dinucleotides, or in short, FAD. Let us start with the first coenzyme, which is coenzyme A. So coenzyme A is an essential, widely distributed thiol-containing cofactor that functions as a primary acetyl group carrier in all cells. So the diagram shows the structures of a molecule of coenzyme A. In our syllabus, you are not required to memorize or to know the structure. So a complete molecule of coenzyme A consists of several essential components. The first component, adenosine diphosphate. Second component, known as pantothenic acid, which is a derivative from vitamin B5, as well as a cysteine amine group. So again, this is extra information for you, and you are not required to remember the structure. In our syllabus, we need to know what is the main role of coenzyme A in aerobic respiration. So coenzyme A functions as a coenzyme by accepting an acetyl group from pyruvate forming acetyl coenzyme A during link reaction. After combinations of acetyl group to coenzyme A, so coenzyme A now deliver the acetyl group to the Krebs cycle. And the acetyl group combine with oxaloacetate to form citrates during a Krebs cycle. And at the end of the formation of citrate, coenzyme A is released from the Krebs cycle during citrate formation and can be recycled so that another molecule of acetyl group can be transported or can be delivered to the Krebs cycle. So this coenzyme A can be reused definitely and continue to form acetyl-CoA in the link reaction. So let us look at this structure of acetyl coenzyme A. So if you look at this, the entire molecule, so coenzyme A actually consists of many carbon atoms. But because in our syllabus we are looking into this acetyl group. So where is acetyl group? So acetyl group actually appear here. This is acetyl group with 2 carbon. Okay, so the remaining molecule, these are the coenzyme A. This is the acetyl group. So entire molecule is known as acetyl coenzyme A. So the role of coenzyme A here is to transfer the acetyl group into Krebs cycle, and at the end, coenzyme A is released here. The next coenzyme we're going to look at this NAD. So NAD is a coenzyme present in all living cells. Okay, so the diagram here show the molecules of NAD. So NAD is known as a dinucleotide because it consists of two nucleotides joined through their phosphate group. So one nucleotide contains adenine base and another nucleotide contains nicotine amide. So don't worry about the structure. You are not required to remember the structure as well as to describe the structure. So NAD is primarily derived from our diet, particularly vitamin B niacin. So common dietary source of niacin includes meat, fish, dairy products, nuts, and whole grain. And our body convert this niacin, okay, and nicotine amides into NAD. Let us focus on the role of NAD in aerobic respiration as is a requirement of our syllabus. So first, what is the role of NAD? NAD acts as a coenzyme for dehydrogenase. So the process of dehydrogenation that occur during glycolysis, link reaction, and Krebs cycle must involve an enzyme known as dehydrogenase. So this NAD now acts as a carrier of hydrogen atom. Be careful, not hydrogen, not proton, but hydrogen atom. They carry hydrogen atom. So in glycolysis, NAD is reduced to form reduced NAD. So NAD actually oxidizes triose phosphate to form pyruvate by accepting hydrogen atom from triose phosphate. Similarly, in the link reaction, the reduction of NAD to reduce NAD allowed pyruvate to be oxidized, leading to the production of acetyl coenzyme A. In the Krebs cycle, again, NAD is reduced, which helps the regeneration of oxaloacetate from citrate. So overall, after this NAD carry this hydrogen atom, so this NAD now deliver this hydrogen atom, which eventually split into the proton and electrons to the inner mitochondrial membrane for this oxidative phosphorylation and also electron transport chain. And this is very crucial for ATP production. So NAD again can be recycled during respiration, provided that oxygen acts as a final electron acceptor. So therefore, NAD can be regenerated in oxidative phosphorylation, and this NAD can be reused. So let us look at this interconversion between NAD and reduced NAD. Again, this is an extended learning, so you are not required to know the reaction. So based on this diagram, you can see that when this NAD is in an oxidized form, when they accept hydrogen atom, it itself is reduced to form a reduced form, which is known as reduced NAD. Let us look at flavin adenine dinucleotide or FAD. So this is a structure of FAD. So FAD consists of two main components. The first component is known as adenosine monophosphate, as well as the flavins mononucleotides connected to AMP or adenosine monophosphate through their phosphate group. So FAD is derived from riboflavin, which is a form of vitamin B known as vitamin B2, which is obtained through dietary sources. So the body converts this riboflavin into FAD through a series of enzymatic reactions in cells. Again, in our syllabus, you are not required to remember the structure as well as to describe the structure of FAD. Our focus is more on to the role of FAD in aerobic respiration. So similar to NAD, FAD is a coenzyme. FAD is a hydrogen atom carrier. So FAD acts as a coenzyme for dehydrogenase again in dehydrogenation reactions during respiration. It acts as a carrier of hydrogen atom, again, hydrogen atom, not proton, not hydrogen gas or hydrogen. So in the Krebs cycle, FAD is reduced to form reduced FAD, and this reduction process helped to regenerate oxaloacetate. Please bear in mind, FAD is involved in Krebs cycle only. On the other hand, NAD is involved in glycolysis, Krebs cycle, and link reactions. So again, similar to the NAD, FAD delivers protons and electrons, or more precisely, delivers hydrogen atom to the inner mitochondrial membrane, and this hydrogen atom splits into protons and electrons. And this is crucial for ATP production in oxidative phosphorylation. And as we know that with the presence of oxygen act as a final electron acceptor, FAD can be regenerated and recycled during respiration, continue their role in the dehydrogenation reaction. So, yep, this is another extended learning, okay, where we look at the interconversion between FAD and reduced FAD. So again, when FAD receives hydrogen atom, it itself is reduced to reduced FAD. With that, we have come to the end of this lesson. I hope you have found this lesson helpful. If you have any question, feel free to leave them in the comment section. See you in the next lesson. Have a great day.